Thank you for agreeing to be part of the Accelerating Biotherapeutic Development and Production for Increased Market Impact of Life-Saving Therapeutics. This is a session at this year's virtual cell and gene therapy meeting on the Mesa. I'd like to introduce myself. I'm Susan DeCosta. I'm the Senior Director for Technical Program Design at Thermo Fisher Scientific. And I'd like to introduce my panelists. We have Nripen, Michael, and Alex, and I'll let each of them introduce themselves. Nripen?
Sure. My name is Singh. I am with Voyager Therapeutics. I lead the biopharm development within technical operations, been with Voyager for more than 1 year. Prior to that, I was at Bristol Myers Squibb and Medipur Sigma. And I'm looking forward to the panel discussion.
Well, welcome, Nripen. Michael?
Hi, my name is Michael Mercaldi. I'm the Senior Director, Downstream Process Development at Homology Medicines. In Homology for about 3 years. And previous to my career, I've worked at Kodiak Biosciences, Merrimack Pharmaceuticals, and MedImmune AstraZeneca.
Welcome, Michael. Nice to have you here. And finally, I'd like to introduce Alex.
Thank you, Susan. I'm Alex Fotopoulos, Chief Technical Officer for Passage Bio. I joined the company July of last year. I oversee CMC, manufacturing, and technical operations. Prior to that, I worked in a gene therapy company, Dimension Therapeutics, which was acquired by Ultragenyx. And prior to that, I had appointments with Momenta Therapeutics and Biogen. Great to be part of the panel. Great to see some of friends like Susan, and great to have a discussion with Monica and Nripen.
All right, so let's get started, guys. So let's start with maybe a more of an icebreaker question. What was the most important thing you learned as you brought or are bringing your product into market?
I can start, Susan, if you don't mind.
Sure, go ahead.
I would say the most important thing that I have learned, specifically with regards to cell and gene therapy, is that as these products move into the later stages of development, careful consideration should be given to producing them at scale. For cell and gene therapy companies, figuring out when to look in the space needed to make these drugs is very critical. We have all heard that there is shortage in capacity right now in cell and gene therapy. There are estimates that it's 5 times now and will be more than 50 times in 5 years, given the fact that 750 trials of cell and gene therapies and almost 30,000 patients were underway as of June 2020. This is just a report that came from McKinsey in August. And the dollars flowing into these companies, it's very critical to build robustness of supply chain to de-risk the clinical trials or commercialization of programs. This is quite interesting to me because I came from big biopharma, and that was not really the issue. But as you can imagine, it's altogether a different place, and manufacturing capacity is going to be quite critical for the success of all the gene therapy products.
Yeah, I'd like to kind of echo that comment as well, is that I think as an industry, especially as in tech ops, we really need to be focusing on scalability. And that's something at Homology we've really focused on a lot. We've been able to prove we can do 2,000 liter production with our triple transfection platform. And we have a scalable purification process using chromatography that can deplete empty capsids as well. To follow on from that, I think the biggest lessons I've kind of learned is you got to keep an open mind. You know, a lot of us have come from recombinant proteins and MABs, and those are so tried and true. But here, there's so much to learn, and we really need to really kind of focus on learning and improvement. And one kind of thing we pride ourselves here at Homology, there's no such things as mistakes, right? It's more opportunities for learning, opportunities for growth, and in the end, we're going to make better products.
That's right.
Absolutely.
I agree with both. And I would say, first of all, we are an early stage company. We are trying to get into the clinic. We're trying to show efficacy with some very novel cutting edge programs. I guess all the panelists, we all work in vivo gene therapy, which means the supply chain is very complex, very difficult. But I would say one of the considerations, at least, or one of the key lessons learned from my side has been the analytics. Without analytics, very hard to do CMC, very hard to talk about scalability. It's very hard to characterize the product. It's very hard to understand how the vector or the product works and how the process impacts it. So analytical development, analytical methods is a significant concern to advance these modalities to commercialization.
Yeah, that's a great point you make. For example, with the MycoSeq kit, right, that Thermo Fisher is putting out there. Now we can bring qPCR on the foreground, and we can talk through, you know, taking a more classical agar-based method to bringing it more into qPCR, kind of staying state of the art with the analytics. So I agree with you, Alex. And you know, scaling up, that's the other problem. So my second question is actually with regards to scale up. And as you scale up your manufacturing for process, there are obviously going to be gaps that you're going to identify. So what are the top 3 or 5 critical elements that you think you should focus on as you scale up? And this could be anything from raw starting materials, could be resins, it could be single-use equipment. I just wanted to hear your thoughts on that and see whether this has caused an impact when you got into the clinic, and how did you solve for that as a result of this?
Yeah,
So I'll
sure.
stop it. Michael,
Yeah,
go ahead.
so I'm not sure if it's like top 3 or 5. I think you have to go back to your kind of fundamentals and your first principles. And really, if you have a well-defined process and a well-defined product, then you're able to have a seamless transfer to your manufacturing. And that's something that we've done a lot here at Homology. We've invested heavily in internal process development. So then we can really understand our scale down models. We can explore mid-scale production, and then eventually we have successful manufacturing. We've been able to supply all of our clinical trial materials, and we have enough material on hand to support all of our expansion as well. And we really think we're able to successfully deliver on the manufacturing front because we've really kind of focused on defining all the process. And that's from, you know, cell line all the way through the end, all the raw materials, all the process parameters. And then to that, understanding the product so we know how it behaves in the process. So in the end, we have successful batches in manufacturing.
That's right. Go ahead, Nripen.
I concur with all the points Michael made. I would say the critical points or elements for scale up of manufacturing for us would be the choice of expression system, standardization of our manufacturing platform, and robustness of the supply chain. I mean, at the end of the day, the goal of our manufacturing process is to ensure manufacturability, and that would mean that it has to meet the desired attributes of your scalable demands based on the indications, your process economics, which is your cost of goods, and at the same time, regulatory expectations as well. So I believe that scale up can be quite difficult, and that's something which I have learned working in cell and gene therapy, because that would very much depend on the system that you're using. So taking those things into consideration, Voyager has selected technologies for our next generation platform that would support these goals. So starting with our SF9 baculovirus expression platform, which we believe can scale up and scale up well, up to 2,000 liter use of single-use equipments and consumables for upstream and downstream processes that include single-use bioreactors, pre-packed columns, media filtrations. All those things which we have learned in the last decade or so in biologics can be easily applicable to cell and gene therapy products. And that can give us a great deal of flexibility, and more than anything would ensure manufacturability in the long run.
I agree. I agree on both points. And actually, if I may, I would add a couple more. I mentioned about the analytics, but I would also add what I think Michael was making a point, but specifically around pilot capability. What I'm talking about is the largest capacity you can run your process before you go to the GMP. GMP has a lot of restrictions, but one of the biggest concerns with gene therapy is material supply for tox studies, material
Right.
supply for method development, material supply for process development.
That's right.
Tech transfer, troubleshooting your biggest, your process at the scale before you go into to make your clinical or CMC enabling lots that you want to make. So these some of the capabilities, and especially the pilot capacity and capability that actually can really link or connect the research side with the development and the development side with the manufacturing. And that enables both the scale up and the advancement of the programs in the CMC overall.
So really taking a quality by design approach, really pushing hard to make sure that you can build out the robustness as early as possible and then be able to scale that up and take it through your process.
Yeah.
So then in that context, right, as you make these changes to your manufacturing process, you obviously want to make them as early as possible. You want to really standardize your platform. You're thinking about ways that you can then use this as your pipeline for all of your pipeline products. Maybe it's a plug and play. You just, replace one molecule with the other. So how do you envision that would happen? Would you take an integrated or a modular approach? Would you use continuous manufacturing? Something that we can learns from the biologics industry. Just kind of postulate on that.
Sure. Would you like me to start, Michael?
Oh, you can go ahead. Yeah.
Okay. Thank you. As Susan, you mentioned, I think there are several lessons, and I'm going to re-emphasize this. I think I heard about it. I used to hear about it. I hear about it. That there are a lot of things that we can learn from biologics, and I can see it now. I can relate to it because I worked in biologics. I think there are several lessons that we can learn and apply them as we have seen with the advancements that we have made in recombinant protein production. We all know how it has evolved over years. That results in very high productivity now. So there can be a lot of similarities we can see, especially in gene therapy manufacturing. So the platform approach would be the way to go. I think we are all striving towards platform development. It would reduce the development timelines, decrease the cost of goods, move the product to the market faster. And it would also give us an ability to switch between batches and products. However, I feel that there are several challenges that we have to overcome to really work with manufacturing platforms, especially for cell and gene therapy, because of the diversity of the capsids, of the viruses that we use. And it's difficult to use a platform when you have so much diversity, right? But taking everything into consideration, I think we eventually would reach a point where we would need some sort of standardization with upstream and downstream processes, and also incorporating some of the new technologies, because we have leveraged some of the things from biologics might not be applicable for cell and gene therapy. So eventually, I would see all those things coming along, and specifically for gene therapy. I feel modular approach is going to be the way, especially with all these new technologies coming in, and hopefully single use coming to a point that you can make a fully single use platform that would fit to your pre-designed modular facilities. To the last point you made, continuous manufacturing, I mean, there's certain ways that you can decrease the cost, right? And make your processes cost effective. Eventually, continuous processing would also come into play. I think there's a need for it more in cell therapy compared to gene therapy as of right now. So I would say it would be implemented sooner in cell therapy and eventually in gene therapy as well.
That makes sense. Michael, why don't you go ahead?
Yeah, sure. So at Homology, we have our own proprietary suite of capsids, and we've actually developed a plug-and-play platform. It took a lot of effort and energy on our Ph.D. and manufacturing teams, but we're there. It's really simply, we're changing out the transgene, and it's pretty much the same exact process from cell line all the way down through the final vial product. That alone has enabled us to really kind of move forward a lot of constructs through our preclinical pipeline, enable for us to supply our clinical trials quickly. And then we believe we'll be able to expand our pipeline very quickly by using this plug and play platform. And in the end, that really helps us for our ultimate goal is getting into first in human, getting these products into the patient's hands as quickly as possible.
That's good.
So we are also trying and we are striving to have a single platform, similar to what actually Michael said. One consideration about the platform here is how flexible the platform is.
That's right. Yeah.
And how you can work with different phenotypes, even different constructs. Sometimes constructs we all trying to have a single-stranded with the appropriate size for the payload of the virus, but sometimes you may have to work with oversized transgenes. You may have complex transgenes that require they have some challenges. So these are part of what makes the sort of the development of a single platform sort of difficult. And trying to have that flexibility, of course, is a consideration. Having said that, also I agree with Nripen about sort of the appropriate platform. You have to see all the platform not in the context of just pure manufacturing technology, but actually within the context of developing therapies. And in our case, we are working on, or the majority of us, we are working in the rare disease space. That's where part of this meeting.
That's right.
And create some interesting challenges for how much time you have, how you can take this platform to commercialization. So you have to consider all of those factors when you develop in order to, make the right choices. Development is all about choices that you have to make and decisions along the way in order to understand how far you want to go with either the manufacturing platform or with the development, in order to bring it to commercialization and bring these therapies to the patients.
That's right.
So just
Go ahead, Michael.
Yeah, if you don't mind.
No, go ahead.
Talk about platforms. Sometimes, in our field, we kind of focus on us, right? Like tech ops. Really what we need to be doing is engaging with our friends in research, right? They design the molecules. They go after the diseases. We enable that value in tech ops, right? So early engagement and then working with them to define not only the process platform, but the product platform is really going to be kind of the key. I mean, this is kind of the antibodies before I got into antibodies, right? Like, that's something I think we're doing. Antibodies. And that's something we're doing here at Homology. We talk to our friends in research, we agree on the right construct, and that's when we're going to really develop these quickly.
Yeah, that's a great point. We're usually focused on the process because that's what we are all doing together right now in terms of tech ops and manufacturing. But the product point of view is something that's really important as well. I just wanted to speak on a point of view from a CDMO perspective, where we would start with the same starting material as part of our platform. So, for example, in the past, as part of Brahma Bio, we didn't have the luxury to do this, where we would have a single cell line that we could then continue to propagate no matter what the client was. Now, in this case, with being acquired by Thermo Fisher Scientific, we're in the process where we can use the CTS VPC cell line. And now we're basically building out the process all the way from thaw going into 200-liter, 4 by 200-liter configurations. We've understood the cell expansion. We've understood how those cells grow. We've built data robustly around that cell expansion, and then being able to get the cells to the right confluence to be completely receptive for transfection complexes is really a critical aspect of what we do here. So here's an example on a CDMO perspective where we can say, here's how we can platform and standardize the process so that it can get a little bit more plug and play with our customers' transfection complexes. But that kind of talking about starting materials brings us right into supply chain. And I wanted to kind of bring up, especially in today's environment, we're dealing with the pandemic and we're trying to move a lot of material, whether it is rapid test kits, whether it's qPCR or serology kits, and now we're going to start moving a lot of vaccine material. It's going to become really difficult, but in general, I would say the supply chain for cell and gene therapy products have been pretty challenging and being able to work around that. So what steps have you taken to mitigate the risk, whether it's a single source vendor, whether it's critical or non-critical raws, consumables coming from regions that might be BSE, TSE prone? You know, what steps have you taken so that you can have all of your raws and consumables available for manufacturing?
Yeah, I can go first on that one. So early on in the early days of the pandemic, we kind of sought out and procured a lot of our high risk raw materials. We made sure, you know, things like plasmids, vials, all that stuff, you know, to make sure we have full continuity for all of our manufacturing operations. And we've successfully done that. We haven't had any interruptions in our supply chain or manufacturing because we kind of had that foresight beforehand. And also during the pandemic, we've been making sure we've had enough supply and been procuring that supply on hand. So we kind of had no disruptions, and we haven't had any during this pandemic, fortunately.
Alex, why don't you go?
Yeah, I mean, we did exactly the same thing. You know, first of all, we worked very closely with our CMO, and we established, you know, key materials. I think Michael mentioned some of those, like vials or certain consumables around the adherent platform that we're using, so we can have, we even purchase raw materials at risk in order to maintain an inventory. And we were able actually to, to spread the risk, primarily because we have multiple programs, and that created enough sort of inventory that we need to carry that actually we can, you know, balance between different programs. We also manage very quickly plasmid supply. This is, you know, we're trying to ensure, and we continue to do that. This is sort of a critical component, at least in our platform, that, you know, we trying to establish a very, you know, dedicated capacity and make sure that we have enough plasmids, not only for what is happening today, but actually as we look further out next year and beyond. One area that has been an interesting area is the CRO testing, and that's something we worked, and we are still working with a number of third-party testing labs. Those labs, they seem to be more sort of, you know, a pinch point right now, and that's sort of where we are putting our focus, primarily establishing the appropriate relationships with these sites.
So are you using a vendor to manage your production, chemicals and the like, or are you doing this in-house with multiple vendors?
At least in our case, we're using a hybrid approach. We are not using a single approach. Depending on the raw materials, depending on the components, including, you know, consumables, depending some of the setups, they may be a different approach to use here, including efforts, our own efforts, how to procure them and stage them.
That makes sense. Nripen?
Sure. Yeah. I mean, I agree with Alex and Michael for some of the measures that they are taking. We are taking as well. The most important thing that I have learned, or we have learned as an organization, is to have a reduced reliance on single suppliers, and then making sure or ensuring that we have manufacturing capacity planned for critical programs with multiple vendors, so that it de-risks our clinical and commercial program. So that's something which we are taking into consideration. Obviously, we have seen challenges because many of the raw materials that we are using for viral vector vaccines are pretty much same as what we use for manufacturing gene therapies. So to identify and validate 2 to 3 suppliers, possibly early in the program, rather than relying on just one, is going to be quite critical. I mean, just for example, single-use bag assemblies have a lead time of more than 6 months now, compared to 3 months we were seeing before. And I don't know whether it's because of COVID situation or it's because of the number of the programs that are going into clinic.
Right.
But it's very critical to work with multiple vendors to ensure the business continuity.
Yeah, I know. I'm assuming all of you are doing suspension technology, so this wouldn't have been relevant to you in terms of the hyperstack shortage that we saw post Hurricane Irma. But that's another example of where, plastic, where, for example, went completely in a situation where you had to actually hold and stock up to be able to make that happen. But also the material that is required for tubing assemblies, that was a situation as well. So kind of thinking through, what are the alternatives in terms of materials that you could use for tubing assemblies and the like, would kind of puts a big burden on all of us in terms of making sure we can source that raw material, have alternative sources, whether it's just the vendors but also the material itself. So that's a good point here. So I wanted to take a little bit of a detour and talk more about partnerships and collaborations that you've developed during your product development. And really ask the question, what have you found to be the biggest help from your outside partners? And how can you promote more collaboration with suppliers? So I'm focusing on maybe vendors, not just the service industry, but also vendors and suppliers for your different raws, consumables, equipment, and the like. And what are the qualities in your partner that you're really looking for? Alex, go ahead.
Sure.
I can go first. Again, I'm going to talk about in the context of relationships with suppliers, with vendors, and service providers. So, clearly the biggest thing that, I think, I'm looking into besides the relationship is, and having a healthy relationship is really the value they bring in. The know-how. It's not just the product. It's not just a service. It's really, understanding, knowing what exactly the offering is, how to support it.
Correct.
We talked about, single-use systems. There is a plethora of information that is necessary, especially in this field, that we need to get information that actually, the suppliers are better suited. It's not just because it's the supplier who provides the product, but they are better suited, they have the better capabilities, and they have a better understanding, and they can provide information that can really enhance not only the value of using the material, the service, but actually enhancing the experience and also the end product that goes to the patient. So for me, and having, working with suppliers that can provide that technical support, that technical know-how, it's extremely important. Of course, there are always the consideration about supply, how they supply the product, how we can continue, the lead times we talked about. But without that part of the technical support, many times the product is really, orphan, and actually falls into the sponsor, if you want, or the user, really to develop a lot of information that typically should come from the vendor. And actually that product becomes much more, more reliable and more brings more higher demand, the more comes with all the supporting information and technical support.
Yeah, that makes sense. Nripen, you wanna?
Yeah, I mean, I could, maybe I will just try to answer that question in the context of what we are doing with our model. So Voyager's current model is based on hybrid manufacturing, which is basically in-house process and analytical development, whereas GMP work is outsourced to external sites, right? So we have a heavy reliance on our partners. It helps us to de-risk manufacturing timelines because multiple programs may compete for capacity and tech transfer resources. So we really want to work with our partners who can give us the capacity, but not just the capacity, the know-how of how manufacturing works, so that we can advance our programs through development lifecycle. I mean, a very simple thing, but quite critical when it comes to products, leveraging GMP quality systems, right, from our service providers to support our critical quality activities. So it's quite critical to maintain that relationship between drug developers, manufacturers, CMOs, and ultimately their suppliers, because that's very critical as well. Alex touched upon it. It's very important to understand our process and product requirements. I think vendors can really help us to provide this expertise on the equipment and various processes, right? Especially for upcoming technologies, enabling technologies that can come both in upstream and downstream viral vector manufacturing. And as the industry would mature, we expect that vendors would play a bigger role and help to improve the productivities further, right? Just like what we saw in monoclonal antibodies in last 3 decades or so. So it's extremely important for us to work closely with vendors, with service providers who can help us to customize our entire platform, because we wouldn't have a better platform if we don't have the solutions that we need for our manufacturing processes.
Yeah. Michael?
Yeah, yeah, I think similar comments. I think I want to take a little bit of a different direction and talk more about the vendor relationship and the service provider. We have great service providers. We've been very happy with them. And they've really helped us develop our products and our processes really well. Something that I think as a field we need to do a little bit more of is collaborating with vendors, right?
Correct, yeah.
And the rationale behind that is we have access to the feed streams and the knowledge that the vendors are just really craving for. You know, a lot of them can't build up the capability to get these feed streams. And I think by having that kind of collaboration, we can then help the vendors develop products that are more tailored towards, you know, AAV production or cell and gene therapy production. And that's something that we have been doing with those small and large players here at Homology. And we feel it's been very beneficial. We've been making great progress with understanding our platform and our products doing that. But as a field, I know we do have a tendency to be a little bit more holding things a little closer. But I do think we need to kind of engage our friends at the vendor companies in order to make better products. So this is going to help us in the end.
Yeah, that makes sense. I definitely think about, you know, when you're thinking about bioreactors, for example, having a single controller that can control different kinds of bioreactor vessels might be something we would be interested in. So, you know, I know Thermo Fisher has the high performer controller that is using the Delta V system as its fallback on the software, but you know, can control a bunch of different kinds of vessels, whether it's Sartorius or Millipore or other kinds of vessels. And this will help to kind of build that kind of you know, relationship with the vendor as you're thinking through, you know, what your current technologies are, but you can then take that to the next level as well.
Yeah, for sure.
So the next question I have is, what type of custom solutions were needed to bring your product to market or get it ready for market? Did you have to customize development in-house? And I know, Nripen, you kind of brought that up a little bit with your previous conversation, or did you go with a partner or a supplier? And it sounds like, Michael, you have a lot more in that context there. And what would you have done differently if you had to redo that again? Nripen, why don't you go ahead and start?
Yeah, so I would say, I think we have, as I mentioned, we have used, or we are using hybrid model, right, to work with our vendors to specifically work on some of these critical raw materials and consumables. So 3 things that come to my mind where we are leveraging the expertise and then working with the vendors would be our custom buffers, for example, for chromatographic resins. And customized bag assemblies. So that really helps to meet our specifications, accelerated our programs. Another thing which is quite critical for our upstream processes is to work with vendors on high performing medias, for example. And that sort of relationship, if you can work with from the very beginning with your partners, is going to really help us to come up with better productivities, right? So you can do a lot of this analysis, amino acid analysis, your vitamin levels, all the critical elements in your media, hopefully to provide better formulations that would eventually increase your productivity. The last thing which I would say is single-use fill finished bag assemblies. They're quite critical. Another custom product that we are working quite closely with vendors. And again, it provides us flexibility to establish these new processes pretty fast. So coming back to the question you were asking, I mean, these are the three examples that I was come up with because I've dealt with them in the recent past. But it's extremely important to have that sort of relationship, almost working in a collaboration with vendors, and to leverage their expertise to accelerate our programs.
You brought up a couple of different points, but what about analytics? Have you had to build some kind of a partnership with a vendor to kind of expand or make more robust some of the analytics that you would use for these processes as well?
Oh, absolutely. I mean, analytics is I think it's, we don't talk about it often, so I'm glad that Alex is touching upon it, but it's a big pain point. Right. And that's something which is quite different from me, from what I used to see in biologics. Now, certainly you are working with 40 assays for your product, and some of those assays are quite difficult, time consuming. So it's extremely important to work with vendors to come up with better technologies, to come up with next generation methods. So to answer your question, yes, we have been working with vendors on that front as well.
That segues straight into Alex there, because that's one of his pet conversations.
Yeah. I appreciate that. So I completely agree. I mean, we are working exactly, I can even sort of go through the same list of all the different aspects that Nripen mentioned. Yes, we are fortunate. We have a collaboration with the gene therapy program at UPenn. So we have access to a lot of the, for example, the analytics, but still we need to work, and we work with a lot of different external parties. And, you know, in my view, this is one of the areas that actually is evolving, is going to evolve very fast. There's a lot of, you know, vendors that are coming up with interesting technologies, and from, you know, new methods all the way to high throughput analytics, which become very, in my mind, a very important tool for development and developing the next, you know, generation or the next commercial process. So all of these collaborations are very important. Having that sort of access to those technologies is very important, and having that those relationships with these vendors is extremely important to understand what is coming down the pike.
Michael.
Yeah, I think I can continue a little bit of the conversation on the analytics, and then I can talk about maybe a little case study on kind of the original question, which is more custom solutions. So I think for us, we've really invested a lot internal. So we have our own internal analytics department, and that has really helped us a lot in that we have dedicated teams that are dedicated to our products full time, that are able to really delve into the products, understand them better, develop better assays. And our assays are working really well. We're able to actually monitor and track the product well, ensure patient safety, and ensure that it works. So we feel that's a key advantage for us, having that internal dedication. Back to the original question, which was like custom solutions.
That's right.
Once again, I'll talk more about internal. We have our own internal manufacturing facility. And a big part of that was we sought out, when even designing that facility, to make it completely single use. And the rationale behind that was we wanted to turn this into a multi-product facility that can churn out a lot of products really quickly. In order to do that, a single use facility is able to reduce any kind of turnover issues, any lot-to-lot turnover issues. And we're able to accomplish this through a lot of great partners and vendors in the single use space. We're very fortunate in the greater Boston area to have a lot of great single use vendors that we can call up, get supplies, to get it to us quickly, and we can work with them very easily to design all these different configurations for all the different products and all the different lot sizes. And I think that's another key advantage for us, is the internal capability, but then also maintaining and building those relationships to keep the internal capability up and running.
That's great. So I would like to kind of start to, wind this down and start asking about the future. So where do you think the future of the cell and gene therapy industry would be, maybe in the next 5 to 10 years? What do you think in terms of technology that's going to accelerate this field? And how can we as manufacturers of service, equipment, raw materials help to influence and align to increase the market access of breakthrough therapeutics? So why don't we start with you, Alex?
Thank you. I don't think it's going to be, first of all, there's going to be a lot of evolution, and it's going to be the same, not maybe the same, but sort of the parallel or the analog here, or is the protein world or the biologics and the mAbs that, it took about 2 decades to advance from, make move from something that it was very unique, very different, multiple different platforms to something that is much more now, I won't call it commodity, but it's really a standardized platform for generating antibodies for different indications. I think the same paradigm is going to happen here. I believe this paradigm is going to be much faster because of a lot of the learnings out of the world of biologics, and actually because there are so many, a number of different technologies that are going to influence that. I don't think it's just going to be a single one. We talked about the analytics, but I can say the same thing can happen with next generation of serotypes or capsids, design of the transgenes. CRISPR technology. I'm not talking about gene editing as much even as a tool for developing interesting assays or methods that basically enable drug development. So in my view, there is not going to be one technology that is going to influence, but many. And of course, vendors and suppliers, they have a significant role. As Nripen said, cell culture media. That's what happened before. I think it's going to happen here. Transfection technologies, or if you go to different platforms, how infection is going to work. And then, of course, downstream processing, which is, there are quite a lot of parallels, if you want, with the protein world. So those things are going to come, in my view, very fast, especially with the investment in the field. In the last, if you look into the last 5, 8 years, there has been a significant investment and continues to be. And there's also a technological evolution that we see already happening. So the next 5 years, I think there's going to be a lot of changes in the field. And there's going to be a very dramatic change from similar to what we saw in the mAbs, but much, much sooner.
Makes sense. Michael, what's your take?
Yeah, I think over the next 5 to 10 years, we're going to see increases in overall productivity and yield. I think that's going to be a given. We're getting there, right? I think the reason why we're going to be able to get there is we're getting a better understanding on the cellular processes that make that package our vectors. And with that understanding, then comes increases in productivity and breakthroughs and everything like that. I think the other thing we're going to see too is more focused on capsid engineering and overall vector engineering, because it's not only the capsid, which we're good at because it's the protein, but also the cargo, the transgene, the DNA inside.
That's right.
We're developing more of those learnings and understandings. We're then going to figure out how to make more potent vectors. And then maybe we don't need as much vector to dose our patients. And then that can mitigate some of the other concerns that are going on in the field right now. So I do think the future is very bright for the field. And I think we're going to make more vector and probably better vector in the next 5 to 10 years. I think the second part of your question about the vendors, I think this is where, similar to what Alex was saying, like we're going to see a little bit more of that kind of mAb trajectory, where I think we're going to see more raw materials tailored, though not for mAbs, but to AAV or cell and gene therapy.
That's right.
So cell media feeds more tailored towards that. And then on the purification side, I think chromatography resins and separation technologies for nanoparticles, because right now a lot of us are repurposing things developed for proteins, and I think that's where, bringing back to like an earlier answer, working with our vendor friends to then figure out like, okay, what are the best separations for nanoparticle technology? and for virus separation. And I think that's where we're going to be seeing in the next 5 to 10 years as well. And then overall, I think it's going to be beneficial for all of us because we're going to make more vector and we're going to make better vector.
That's a very good thought, yeah. Nripen?
Sure, yeah. In addition to just what Michael and Alex said, I would say I think the number of indications that cell and gene therapies would target would increase, right? Until now, we have been primarily focused on ultra rare diseases or rare diseases, but as we move to some of the other indications where the requirements are high in terms of VGs that we need specifically for gene therapy, it would be very important to improve the productivities, right? So I totally agree with Michael that vector engineering is going to be quite critical. Another thing that comes to my mind is the creation of stable cell line, producer cell line. I think it would really evolve it would lead to a cell line like CHO that we have for biologics that most of the end users or biotech companies would eventually work with. I mean, transient transfection, no offense to the folks who actually use it, is inherently variable. So the
and
only
the cost
solution,
of goods is high as well, right? Yeah.
Absolutely. I mean, plasmid DNA cost is crazy high. But it would eventually lead to a potential solution from the engineering and the use of producer cell lines. So that's one thing which I think is absolutely going to happen. Obviously, upstream and downstream processes would become much more productive. We would have solutions for both upstreams and downstream technologies. And the last thing that I believe is going to happen is the collaboration between the disciplines. It would happen more and more because that's the only way we can make these technologies or cell and gene therapies accessible to a large population at an affordable price. So overall, I would say the improvements required a coordinated effort combining both biological and engineering expertise.
Yeah. So one thought that came to me as you guys were talking about this was, you know, we're never going to walk completely away from transient transfection, especially when you're trying to get first in human, right? You're not going to be able to say, I can make a producer cell line really quick. And really, what's the benefit of making a producer cell line if it's not going to be a clinical trial that's efficacious? So kind of thinking about segueing back into my previous question on plug and play, whether that would mean you would make actually more packaging cell lines that would, you know, take account of the capsid engineering and the other aspects that you talked about, and then just plug and play with a transient transfection with the transfer plasmid, and in parallel start talking about building out that producer cell line, right? Yeah. Some really good thoughts here. I really think this was great to kind of think about it in that context. But that kind of brings me to today's environment. We're dealing with a pandemic the world over. We're trying to solve for a COVID-19 situation, which would not necessarily go away just magically. There's going to be some kind of intercession needed, whether that's a vaccine or other ways that we can, you know, get our public health there. But clearly, business as usual has been impacted. It's not the old normal. We're trying to walk through a new normal. We're trying to come up with ways that we can, you know, continue with working, manufacturing cell and gene therapy products. So my question here is, how has the situation impacted your progress, the way you work, and what are some of the positive experiences you've had as a result of the lockdown and the COVID-19 situation? Nripen, you want to start?
Sure, yeah, no problem. Thank you. I would say the manufacture and the delivery of these treatments, right? Research and clinical development and commercial operations are three areas within the cell and gene therapy industry that have been impacted the most, or I should say disrupted the most. And that's very much applicable to us as well. So as I mentioned earlier, there have been instances where supply shortages have caused problems. We have experienced long delivery times for certain single-use bag assemblies or critical reagents for our analytical methods, such as ELISA antibodies, and that came maybe due to the closed borders, reduced air traffic, delayed flights. I don't know, I'm just speculating, but these must be some of the reasons that must have impacted the progress.
Right.
And it has impacted our programs as well. So I would say the biggest thing that we learned is to ensure that we have a good supply chain resilience system built in our system. And very important to work closely with vendors and CMOs to address these bottlenecks in our current process. I mean, we already knew about some of these shortcomings, some of the bottlenecks in cell and gene therapy. Now it has actually come to the forefront even more, and then helped us to come up with a strategy that would eventually help us for the success of our programs. So I think that was the biggest takeaway for me from this COVID situation.
Any positive experiences as a result of it as well?
We know how to work in this environment and know how to work quite effectively. So that's definitely a very positive feedback.
Alex.
Yeah, it's been an interesting time with the COVID-19. In our case, actually, it's an interesting story because we had to build a company while under these sort of considerations. And actually, within the last sort of six months or more, we were able to double, triple the size of the company and actually bring more people on board than actually when we were working face to face. And that actually had to change a lot of things because now you have a very different engagement, very different communication model. And that actually had to happen not only internally with how we work, and especially with new people, but actually even with our external partners and our CMOs or our manufacturing partners. You have to change the communication. You cannot follow the same paradigms as they were before. The positive thing has been the fact that actually people are more poised and more dedicated, and they value the time and the communication. And that's what actually I have found as a positive thing. And during this time, it's really that people, how dedicated they are. It's not only working remotely. You have to appreciate the employees. They have families, they have children, they have elderly in the house. So it's not just, just working from a distance.
That's
so,
right. Yeah.
Having said that, all the people, especially in this space, because of sort of the great cause that we have, the great we are all dedicated to the patients. You can see really the good side of people, and you can see how people are willing to go really the extra mile and make things happen. And we saw the same thing, by the way, even with vendors and suppliers. It's not just speaking about our employee base.
That's right. Yeah. Michael.
Yeah. So for us, we've been able to continue our operations the whole time throughout the whole pandemic. We were kind of fortunate. Our governor deemed biotech as an essential business, so we were able to keep our operations open this whole time. And how we were able to do that is, one, we want to make sure everyone was safe, right? So we had staggered scheduling for everyone, really strict social distancing in the facility. Unidirectional workflow, proper PPE, extra PPE for people that wanted it. And that way we were able to keep everyone safe. And then in order to keep everything going, kind of what we talked about earlier, we made sure we ordered a lot of our key raw materials beforehand. So before the supply chains got interrupted, to maintain those continuous operations. I think for positives, I think, going through a crisis kind of makes people a little closer with each other and a little stronger. And I think, you do it through kind of venues like this, but you have maybe a little bit more in-depth conversations with your kind of coworkers and colleagues. And
that's right. Yeah.
Through this that maybe you didn't have the time to do in person. And I think that's something that I've noticed, and I think a lot of the people have noticed here that we've been able to take away with during this difficult time.
Yeah, thank you. So we're coming to the end of our, I thought it was a very productive discussion here. And I wanted to kind of close us out with any kind of concluding remarks or suggestions or comments that you had. Alex, why don't we start with you?
Yeah, I want to say that this is a great forum to have to exchange information. Clearly, we want more of those things. We need more of those things. I think there is a lot of how to say resources in the industry that can be, not in the competition in the sense we all compete. We all trying to do, we are in this space, but there are things that we can share as sort of companies, individuals, even sort of SMEs, if you want, that we can advance them, not only the field, but actually, advance these therapies and bring sort of these therapies to people with a great need. And, these are some of the things, hopefully we can sort of have more, and these times have been very challenging to bring, different companies, different people in one place. So I think having those type of forums, having these type of discussions, and actually this dialogue, I think can help with, many topics that they are challenging for all companies, especially regulatory or, how to approach certain problems or situations. So I think this is something that I'm hoping and I'm looking forward and, to seeing more and, the collaboration beyond just the boundaries of a company or a university.
Any other parting thoughts, Michael or Nripen?
I would just quickly say that, it's an exciting place to be in, the cell and gene therapy industry. And as Alex mentioned, I think we need to think beyond the collaborations just with the vendors and our manufacturing partners. As I think we need to have more collaboration between companies, exchange more information. That would really help us to learn at a faster speed and then accelerate these programs so that we can get them to the patients.
Yeah. I think for my parting thoughts, I think first, Susan, I'd like to thank you for moderating this. Thank Biotherapeutic. It's been a great conversation. And also want to thank the Meeting on the Mesa organizers for having a venue like this. Really shows that manufacturing is important for bringing these therapies. And I think in a meeting like this, to have a discussion around manufacturing, I think is great. I like the question that you posed a few questions ago about the next 5 to 10 years. And, I kind of feel we're at the beginning of the understanding and, we're able to supply, we're able to get into the clinic, we're able to see what these therapies, the potential is. I really think this field has such a bright future, even the next 5, 10, 20 years, where we're just going to see a lot more cures and a lot more manufacturing advances in order to get these cures to patients. So I'm really happy to be working here. I'm really happy we're talking about this at this meeting. Yeah. And, I think it's all great and exciting.
Yep. Yeah, Michael, I echo your sentiments. So Michael, Alex, Nripen, it's really great that you were willing to be part of this panel, and your thoughtful commentary today was really inspiring and really exciting. So I want to thank all three of you for being part of this panel. And Michael, like you said, I want to thank the organizers, ARM, and Thermo Fisher Scientific for making this panel possible. Thank you.
Thank you.
Thank you.